So think about if you break your arm, right? An X-ray gives you this really clean binary answer. It's broken or it's not broken.
Right, yeah. It's right there on the screen.
Exactly. The doctor points to this jagged white line, you get a cast, and you go home.
Yeah.
But if you're diagnosed with breast cancer, the image under the microscope is really just the beginning of this massive, incredibly complex, and honestly, very expensive biological mystery.
Yeah, because biology rarely gives us a clean binary answer. I mean, the deeper we look into oncology, the more we realize that a single diagnosis is, well, it's actually an umbrella term for a whole multitude of different cellular behaviors.
Which is exactly why we're dedicating today's deep dive, which, by the way, we curated specifically for you, to a massive bottleneck in how we currently treat this disease.
It's a huge issue.
It really is. So we are looking at a 2025 research paper by 𝗵𝗵𝗏 and colleagues, and they're tackling a problem that literally dictates the survival chances of millions of people, which is how do we rapidly, accurately, and affordably figure out exactly what kind of breast cancer a patient has?
Right. And to understand the solution they're proposing, we really first have to understand that clinical roadblock you mentioned. Treating breast cancer today relies heavily on something called the PAM50 gene signature.
The PAM50, right?
Yeah, it's a diagnostic tool that looks at the expression levels of 50 specific genes within the tumor. So based on how those genes are behaving, the cancer is categorized into one of several distinct molecular subtypes, the main ones being Luminal A, Luminal B, HER2 enriched, and Basal-like.
And just to ground this in reality for you, knowing that subtype is everything. Like if a patient has a Luminal A tumor, it's typically hormone receptor positive and it grows relatively slowly.
Exactly.
So the oncologist knows they can likely use hormone therapies.
Yeah.
But if it's Basal-like, which often overlaps with triple-negative breast cancer, it's much more aggressive and it just doesn't respond to those same hormone treatments. The subtype literally dictates the battle plan.
It does, but the issue is actually getting that battle plan. Identifying the PM50 subtype traditionally requires RNA sequencing.
Which is not simple.
Not at all. I mean, you have to extract genetic material from the biopsy tissue, run it through sophisticated sequencing machines, and then have computational biologists interpret all that transcriptomic data. It requires highly specialized laboratory infrastructure. It takes a lot of time. And crucially, it is extremely expensive.
Meaning a world-class research hospital in a major city can do it without blinking, but a smaller clinic or, a hospital in an under-resourced area simply cannot.
Exactly. They are forced to rely on slower, less precise surrogate markers. So the fundamental question this research asks is, well, what if we could bypass the RNA sequencing entirely?
And this is where the AI element comes in.
Yeah.
Because almost every hospital in the world, regardless of their budget, produces standard biopsy slides. They slice a tiny piece of tissue, put it on a glass slide, and they treat it with a chemical stain called H&E hematoxylin and eosin.
Right, and the mechanics of that stain are just incredibly reliable. Hematoxylin binds to the DNA and RNA in cell nuclei, turning them a dark blue or purple. And the eosin targets proteins in the cytoplasm and the surrounding connective tissue, turning them pink.
The classic pink and blue slides.
Exactly. It creates this vivid structural map of the tissue morphology.
So the promise of this paper is training an artificial intelligence to look at those cheap, universally available pink and blue structural maps and accurately predict the underlying 50-gene PM50 molecular subtype.
Which is wild when you think about it.
It is. If you can do that, you essentially democratize precision oncology. You turn a basic digital image into a high-end molecular assay.
Yeah, on paper, it sounds revolutionary. But in practice, feeding those images into a neural network creates this monumental computational hurdle. It's what we call the where do I look problem.
Because of the size of the images.
Yes, you have to conceptualize the sheer scale of a digital biopsy slide. We call them whole slide images, or WSIs. When a standard tissue slide is scanned at a high resolution, like 40x magnification, it results in an image containing literally billions of pixels.
Billions. So an AI cannot just swallow a billion-pixel image at once. I mean, it would instantly max out the memory of even the most powerful graphic processing units. The system would just choke.
It absolutely would. So to get around this, computational pathologists take that massive image and slice it up into a grid of tiny, digestible squares. The researchers in this study extract patches of tissue that are 512 by 512 pixels.
Okay.
Then they mathematically compress those patches down to 224 by 224 pixels, mostly because that is the standard input size that most visual neural networks are designed to process.
Okay, so you take one glass slide and you essentially chop it up into confetti. How many individual patches are we talking about per slide?
Well, after they use some basic filtering to throw away the blank white background where there's just no tissue, a single digital slide yields roughly 10,000 individual patches.
Wow, 10,000 images for one patient. Now, standard AI models usually handle this by just sampling randomly, right?
Right.
They throw a dart at the board, grab a few hundred random patches, and feed them to the network.
Unfortunately, yes, that's the standard.
But thinking about the biology here, that seems like an incredibly risky strategy. It feels like, Well, like trying to review a sweeping 3-hour epic movie by closing your eyes and randomly selecting 20 different 10-second clips.
That's a perfect analogy because if you do that, the odds of you actually seeing the climax of the film or even understanding who the main characters are incredibly low.
Right. You're mostly going to get shots of an empty hallway. Right. Or like an extra drinking a coffee in the background.
Yeah.
You completely miss the plot.
And in a clinical setting, missing the plot is dangerous. If you feed an AI unfiltered, randomly selected patches of tissue, you are flooding the system with biological noise. Out of those 10,000 patches, thousands of them are histologically redundant. They might show healthy fat cells or normal connective tissue or benign structures that have absolutely zero diagnostic value for predicting a complex gene signature.
So you are wasting the AI's processing power analyzing background extras.
Exactly. And worse than wasting power, you are actively degrading its discriminative ability. If the neural network is spending the vast majority of its training time looking at irrelevant tissue, it's going to struggle to learn the subtle, complex morphological patterns that distinguish, say, a fast-growing basal-like tumor from a slower luminal A tumor. Its vision just becomes blurred by useless data.
Okay, so we clearly need a way to find the main characters. We need a filter. And this paper tackles this in 2 stages. The first stage might actually be my favorite part of the entire methodology because they essentially teach the AI how to admit when it's confused.
Yes. They are quantifying the AI's epistemic uncertainty.
Epistemic uncertainty. Break that down for us.
Sure. So epistemic uncertainty arises when a model lacks knowledge about the data it's seeing, meaning the model's own internal parameters are unsure because it just hasn't seen enough similar examples during its training. The researchers use a really fascinating mechanism called Monte Carlo dropout, or MC dropout, to measure this.
Okay, wait, so how do you force a machine to quantify its own ignorance?
You test its consistency under deliberate impairment.
Deliberate impairment.
Yeah. They take a single 224 by 224 pixel patch and pass it through the AI's classification network. But they don't just do it once. They pass that exact same patch through the network 20 different times.
Okay.
And during each of those 20 passes, the system randomly disables or drops out 50% of the neural network's connection.
Wait. They turn off half of the AI's brain, seemingly at random, 20 separate times.
That's exactly what they do.
I mean, I have to push back on the practicality of this. If I have a piece of software that takes 20 times longer to load because it's constantly double-checking itself by giving itself, like, localized amnesia, I'm throwing that software out. Doesn't this completely bottleneck the system?
Well, it's a valid concern regarding the computational overhead. Performing 20 stochastic forward passes does linearly increase the inference time for that specific step.
Yeah.
But you have to look at the scale of the trade-off. Running a tiny single patch through a network takes fractions of a millisecond. What they're buying with those milliseconds is an incredibly powerful diagnostic shield.
A shield against the noise we talked about earlier.
Precisely. By randomly turning off half the network's internal pathways, you are forcing the AI to analyze the image using different combinations of its acquired knowledge. If a patch contains highly salient, unambiguous cancer features, the AI will consistently predict the same molecular subtype, regardless of which half of its network is currently disabled.
Oh, I see.
Yeah, the diagnostic answers across those 20 passes will have very low variance.
So it's certain. But if the patch is just, a blurry piece of connective tissue or a bizarre artifact from the glass slide,
then the AI doesn't have strong structural features to rely on, so it starts guessing. And because its active internal pathways keep changing with every single pass, its guesses will vary wildly. Pass number 1 says Luminal A, pass number 2 says HER2 enriched, pass number 3 says Basal-like.
So the variance spikes.
Exactly. The variance spikes, and the algorithm flags that specific patch as having high epistemic uncertainty, and it actively discards it.
Wow. So it throws the garbage out before it can contaminate the downstream analysis. It's essentially learning to say, hey, I don't have enough information to make a call on this tiny square, so I refuse to include it.
Which is a profound form of diagnostic risk management. It forces the final decision-making engine to only look at high-confidence regions of the tumor.
Okay, so we've run the Monte Carlo dropout. We've tossed out the patches where the AI is basically guessing. But throwing out the garbage doesn't automatically mean what's left is a cohesive, efficient data set?
No, it does.
Because even after dropping the highly uncertain patches, you still might have thousands of high-confidence patches left over. And processing all of them is still way too slow for a rapid clinical tool.
That is the second major hurdle. You need to distill those remaining thousands of patches into a really small, elite subset. And to do this, the researchers bring in a mathematical concept that actually mimics biology. They use an evolutionary algorithm called NSGA-II.
NSGA-II.
Yeah, that stands for Non-Dominated Sorting Genetic Algorithm 2.
When you say it mimics biology, we are talking about actual algorithmic evolution here, right? Like survival of the fittest.
Quite literally, it treats the selection of the perfect patch subset as a multi-objective optimization problem. So instead of just trying to maximize one single variable, the algorithm simulates an evolutionary process to balance 4 complementary goals simultaneously.
Let's unpack those 4 goals, because I feel like this is where the genius of the curation really shines.
Absolutely. The first goal is diversity. The algorithm is looking for morphological heterogeneity. It actively selects patches that look structurally different from one another, ensuring the final subset represents the entire landscape of the tumor, not just one concentrated area.
Makes sense.
The second goal is informativeness. It seeks patches with high future saliency, basically regions that are dense with complex cellular structures and rich biological data.
Okay, that's 2.
The third goal is compactness. It wants to achieve the first 2 goals using the absolute smallest number of patches possible to minimize the computational burden.
Right, keep it efficient.
And the fourth goal is reliability. It builds directly on our previous step, heavily favoring the patches with the absolute lowest predictive uncertainty scores from the Monte Carlo dropout phase.
You know, listening to those 4 metrics, diversity, informativeness, compactness, reliability, it sounds less like we are doing computational biology and more like we're, I don't know, assembling a crew for a heist.
A heist. I can sort of see the parallels. How so?
Well, think about it. If you're going to rob a high-security vault, you don't just hire the 5 best safe crackers in the world, right? Because if your entire team only knows how to crack safes, who's hot-wiring the getaway car? Who's looping the security cameras? A homogeneous team fails. You need a variety of skills, which is the algorithm's first goal. Diversity.
But you also can't just hire random people off the street to achieve that diversity.
Exactly. You want elite professionals, hackers who write custom code, drivers who used to race professionally. You need rich, high-level skills. That's your second goal. Informativeness.
And compactness.
You don't want a crew of 50 people. A 50-person crew is a complete logistical nightmare, and you got to split the money way too many ways. You want a small, lean team. Compactness.
Okay, I see where this is going.
And finally, reliability. You want professionals who don't panic when the alarm goes off. You want people whose skills perform consistently under pressure, completely devoid of uncertainty.
Well, when you put it like that, yeah, the genetic algorithm basically operates as the mastermind assembling that percent's crew.
Right.
It starts by generating random populations of different patch combinations. Then it grades each combination on those 4 heist metrics. The subsets that perform poorly are killed off, and the combinations that score highly are allowed to breed, meaning the algorithm mathematically crosses their best traits over into a new generation, introducing slight random mutations to explore new possibilities.
Generation after generation, iterating over and over.
Until it converges on the mathematically optimal subset, and the scale of reduction is just staggering. The algorithm takes that initial slide of roughly 10,000 patches and boils it down to an elite crew of just 500 patches.
Right.
That's an approximate 95% reduction in data. And because it's so mathematically efficient, it performs this complex evolutionary selection offline in about 3 minutes per slide, using a standard GPU.
Just 3 minutes to sift through billions of pixels and find the 500 most critical squares of biology. That's incredible. And from there, these elite 500 patches are fed into a ResNet-18 architecture, right?
Yes. ResNet-18 is a well-established convolutional neural network. In this pipeline, it acts as a feature extractor. It isn't inherently looking for cancer at this stage. It's analyzing the pixels to identify edges, textures, cellular densities, and complex geometric patterns, converting that visual information into mathematical vectors. And those vectors are what finally get classified into the 4 𝗶𝗴𝗵𝗹 molecular subtypes.
Okay, so we have the theory, we have the filter, we have the evolutionary algorithm. But the real world of oncology doesn't care about elegant math. It cares about diagnostic accuracy. Does this highly curated data actually work when put to the test?
It does. To validate the framework, the researchers deployed it against 2 entirely separate datasets. The first was their internal dataset, known as the TCGA-BRCA cohort. This was the environment where the model was largely trained and fine-tuned.
And what were the numbers on that?
On the internal data, the performance was stellar. The model achieved an 89.95% accuracy across the 4 subtypes and an area under the curve, or AUC, of 0.9841.
Wow, an AUC of 0.98 is essentially a masterclass. That means the model has a near-perfect ability to distinguish between the different classes. It proves the foundational concept. You absolutely can extract molecular-level insights from standard, cheap pink and blue slides.
But of course, the true test of any medical AI is generalizability. So they took this model and tested it on an external data set, the CPTAC-BRCA cohort.
Unseen data.
Completely unseen. These were glass slides from entirely different hospitals prepared by different technicians, completely unseen by the AI. And on this external data set, the accuracy dropped to 77.92% with an AUC of 0.8823.
Okay, so it dropped from roughly 90% down to 78%. Feels a bit jarring. I mean, if I'm an oncologist relying on this, a 12% loss in accuracy gives me pause. Is this a flaw in their algorithm, or is this just an inherent reality of medical imaging?
It really highlights a pervasive challenge in artificial intelligence called domain shift. We have to remember how a neural network sees an image. It doesn't perceive a pink cell. It processes a massive matrix of numbers representing pixel intensity.
So even the slightest variation completely alters the math.
Consider the logistics of 2 different hospitals. Hospital A might leave their tissue sitting in the eosin dye for 30 seconds longer than Hospital B. Or they might use a digital slide scanner from a different manufacturer that captures light slightly differently. To a human pathologist, the slide just looks a tiny bit darker pink. The human brain instantly corrects for it.
But to the AI, the underlying mathematical matrix is wildly different. It's like training a facial recognition model entirely on high-contrast professional portraits and then asking it to identify people using blurry, low-light security camera footage. The domain has shifted.
Exactly. And the researchers anticipated this. They actually applied a mathematical technique called Marchenko stain normalization before feeding the slides to the AI.
What does that do?
It attempts to map the color distribution of any new slide to a standardized reference template. It's a way of forcing all the images to share the same baseline filter, but normalization can only do so much. The tissue itself is still inherently different.
You know, what's deeply revealing is looking at where the model struggled on that external data, because the accuracy didn't drop evenly across the board, did it?
No, it didn't.
The predictions for the Luminal A subtype stayed remarkably stable, barely dropping at all. But the accuracy for the Basal-like and HER2 enriched subtypes took a much harder hit. Why the disparity there?
Because it comes right back to the underlying biology. Basal-like and HER2 enriched tumors possess significantly higher histological heterogeneity. They are more chaotic, more aggressive, and their physical structure under a microscope just varies wildly from patient to patient.
So you have a tumor that already lacks consistent visual patterns, and then you multiply that chaos by the domain shift of a new hospital's dye and scanning equipment. The AI is trying to find a needle in a haystack, and suddenly the color of the hay has changed.
That is exactly it. But it's crucial to contextualize this. Maintaining a 78% accuracy on an external, totally unseen dataset for a highly complex four-way molecular classification using only structural images, that is a massive leap forward over previous methodologies.
And the final nail in the coffin, the absolute proof that their specific method is the reason for this leap, is found in their ablation study.
Yes. The ablation study is where the elegance of their design is truly proven. In computer science, an ablation study involves systematically ripping components out of your system to see if the whole thing collapses.
Right.
It proves whether your fancy new algorithm is actually doing the heavy lifting or if it's just window dressing.
So what did they rip out?
They took their entire system and they removed the NSGA-II genetic algorithm. They fired the mastermind. Instead of evolving the perfect elite crew of 500 patches, they just fed the neural network the standard, unfiltered, random selection of patches, the raw noise.
And what happened?
The model's accuracy catastrophically plummeted. It dropped from that internal high of near 90% all the way down to 64.21%.
Oh, wow. A near 26% drop in performance simply because they stopped curating the input.
This is the core insight of the 𝗺𝗫𝗄 paper. It empirically proves that in the realm of deep learning, what the AI looks at is just as important, if not more important, than how it looks at it. You can deploy the most sophisticated, high-parameter neural network in the world, but if you feed it noisy, uncurated, biologically irrelevant data, it will fail. The curation is the catalyst for accuracy.
So bringing all of this advanced computational theory back to you, the listener, why does understanding this specific framework matter? Because this isn't just an abstract math problem. This represents a highly scalable, imaging-based replacement for molecular testing. This framework proves that a small rural clinic, armed with nothing but standard chemical dyes, a basic digital scanner, and an internet connection, could potentially extract the exact same precision oncology insights as a billion-dollar research facility. It is a roadmap to leveling the playing field in cancer treatment.
And stepping back to look at the broader implications of artificial intelligence, this methodology raises a deeply fascinating question about how we design the systems of the future.
Oh, where are you taking this?
Think about the very first step we discussed, the Monte Carlo dropout. The AI systematically testing itself, identifying its own ignorance, and actively discarding the data it feels uncertain about. We live in an era currently plagued by AI hallucinations where large language models confidently output completely fabricated information because they just don't know how to say, I don't know.
Yeah, the arrogance of current AI is one of its biggest flaws.
But this paper demonstrates a system specifically engineered to quantify its own epistemic uncertainty. It is hardwired to refuse to make a decision based on bad data. It begs the question, as machine learning integrates further into high-stakes environments like medicine, perhaps the most valuable feature an AI can possess isn't its raw computational power or its vast intelligence.
Right.
Perhaps its most crucial feature is its capacity for self-doubt.
That is a phenomenal thought to leave on. The idea that true intelligence, artificial or otherwise, requires a fundamental awareness of the limits of your own knowledge.
It really is. And it brings us right back to that complex diagnostic landscape we started with. Biology is messy, it's chaotic, and it rarely gives us a clean line on a screen. And as we build the tools to navigate that complexity, maybe the best system isn't the one that confidently claims it knows everything. Maybe it's the one that knows exactly when to throw out the garbage and ask for a clearer picture. Well, thank you for joining us on this deep dive into the 𝗺𝗫𝗄 et al. Paper. We hope unpacking these mechanisms has been as illuminating for you as it was for us. Until next time.